Welcome to Inert Chemistry. In today's video, our topic is DNA replication. First of all, we will see that what is DNA replication. The process of copying the DNA is known as DNA replication. We can say that the replication of DNA is semi-conservative. It means that during the replication, one old strand or parental strand is used and only one new strand is formed. This is old strand that comes from parental DNA and a new strand is added in it. So we can say that DNA replication is semi-conservative. What are the different steps that are involved in DNA replication? The first step is the recognition of origin of replication. Origin of replication को पहचानना. Our DNA molecule is a long strand, it is a long molecule, and we cannot start the replication from anywhere. There is a specific origin of replication. So first of all, origin of replication is identified. The second is the unwinding of DNA molecule. The third is the formation of leading strand and lagging strand. The fourth is removal of RNA primers. The fifth is proofreading and DNA repair. And at the end, there is coiling of two new DNA molecules, or we can say that there is formation of two new DNA molecules. So in this slide, we will see the various enzymes that are involved in DNA replication. The first enzyme are helicases. They unwind the double-stranded DNA by breaking the hydrogen bonds present between them and thus creating a replication fork. The second enzyme is primase. This generates the short RNA primer along the DNA template. RNA primer, it is actually a sequence of 10 RNA nucleotides that is formed along the DNA template, providing the starting point for the DNA polymerase to initiate its replication. The most important enzyme that is involved in DNA replication is DNA polymerase. It catalyzes the addition of nucleotides to the growing DNA strand. Means it is actually a growing enzyme that makes your DNA. It is of many types, DNA polymerase 1, 2 and 3. But the most important role that is played by it is DNA polymerase 3. It elongates the new DNA strands in the 5 to 3 direction. While the DNA polymerase 1, it removes RNA primers and replaces them with DNA nucleotides during the DNA repair. Single-stranded binding proteins. They stabilize the separated DNA strands to prevent them from rejoining. When helicase unwind the DNA, then two single-stranded DNA strands are formed. So in order to prevent them from rejoining with each other, the single-stranded binding proteins are used. Ligase. They join the Okazaki fragments on the lagging strand by creating phosphodiester bonds between the DNA fragments. Topoisomerase. They help relieve the tension in the DNA strand generated by the unwinding process by cutting and rejoining the DNA strands. Means it helps to relieve the tension in DNA in single DNA strands. Exonucleases, they are involved in proofreading and repair by removing incorrectly paired nucleotides during the replication. These are the 7 different important enzymes that are involved, although there are some other minor proteins are also present. Remember these two conditions for DNA polymerase. The first condition is that DNA polymerase it cannot synthesize DNA on its own, or it cannot synthesize the start the synthesis of DNA on its own. It needs an RNA primer. What is an RNA primer? It is actually the sequence of 10 nucleotides. The new strand that the DNA polymerase form, its direction is always from 5 prime to 3 prime. So here we see about the origin of replication. The first step during the DNA replication is the recognition of the origin of replication. It is done by a complex. In case of prokaryotes, there is only a single origin of replication, but in case of eukaryotes, there are multiple origin of replications present. The origin of replication is actually a repeated sequence of adenine and thymine nucleotides. You know that in DNA, adenine, there is double bond between adenine and thymine. So the repeated sequence of adenine and thymine is first an enzyme complex search the part of DNA in which there is a repeated sequence of adenine, thymine, adenine, thymine, adenine, thymine nucleotides. And from that point, the DNA replication gets started. So here in this slide, we will see a detail of DNA replication. Remember that the first enzyme that is involved, it is helicase enzyme. This helicase enzyme is also called unwinding or unzipping enzyme. It unwind or unzip your DNA molecule by breaking the hydrogen bonds present in these nucleotides. And the second thing that you remember for helicase is that it do not do all this. It do not do the unwinding of this whole DNA molecule at once, because it requires much energy for this. It done the uncoiling of DNA molecule gradually and when it is needed, or when the previous replication is completed. When these single strands of DNA are formed, then single strand binding proteins are attached to it, so that it prevents them from so that this strand do not recoil again. Similarly, this topoisomerase, it is the enzyme that reduces the tension in these single stranded strands of DNA. The main enzyme that plays its role, it is DNA polymerase, especially your DNA polymerase 3. This DNA polymerase 3 is having two basic conditions for its working. You know that these two basic conditions are that the first condition is that it needs a primer. You know that what is primer? A primer is actually a sequence of 10 RNA nucleotide bases that was added by DNA primase enzyme. You know that DNA polymerase do not start the synthesis of or do not start the replication on its own. It needs an RNA primer, and it was added by DNA primase. And the second condition is that the new strand that our DNA polymerase formed, its direction is 5 prime to 3 prime. It means that the old strand on which it works, it should be in 3 prime to 5 prime direction. The two strands of your DNA, they must be anti-parallel to each other. This is your old parental strand that is in 3 prime to 5 prime direction. Your DNA primase add a primer here. This is your primer, and then the DNA polymerase do its work and elongate this primer in and form the new strand in 5 prime to 3 prime direction, means anti-parallel to the parent strand. This strand that is formed continuously without any obstruction, it is called leading strand, while this other strand, it is formed in discontinuous manner, because in this case the parent strand is the polarity of this parent strand is the polarity of this parent strand is here I have right, this is the 5 prime end and this is the 3 prime end, and the new strand that is found, it started from here. Here a primer is added by DNA primase enzyme and it started the synthesis of your DNA in this direction from 5 prime to 3 prime direction, anti-parallel to the parent strand. But and you know that here is your RNA primer is working. We see from this diagram that here your primer was added. This is your RNA primer. It is added here and your DNA polymerase is synthesizing the new DNA strand in this direction from 5 prime to 3 prime direction, anti-parallel to your parent strand that is in 3 prime to 5 prime direction. It do in this direction, but your RNA primer, it is working here. It unwinds more DNA behind this, but our RNA polymerase is working in this direction and the unwinding is occurring in this direction. So this RNA primase, when he see back, he looks back, so he says that, oh, I am working in this direction, but more unwinding occurs behind me. So this DNA polymerase, it jumps back again. A new primer is added somewhere here, and then again he started the synthesis of a new segment of DNA in 5 prime to 3 prime direction, and again it jumps back because your helicase enzyme working in the in back of this DNA polymerase. So your DNA polymerase comes back again and again to synthesize. So as DNA synthesis do not occurs in a continuous manner like in this case. In this case, you know that the direction of unwinding and the direction of synthesis is in the same direction, so it forms continuously without any issue. But in this case, it is formed in discontinuous manner. Only small fragments are formed. Here a primer is added and then a small fragment is formed, then again a small fragment is formed and a primer, then again a small fragment is formed and there is a primer. So between the various primers, small fragments are added. These small fragments, they are called Okazaki fragments. And when these small when this replication is completed, then these small RNA primers are removed from this, and by the help of DNA ligase enzyme, these Okazaki fragments join with each other. You know that DNA ligase is an enzyme that form the phosphodiester bonds between the Okazaki fragments and join them. This DNA polymerase is itself having the ability to recheck all the procedure. You know that this DNA polymerase, it is responsible for this whole process, so it can recheck all the DNA replication process and it sort out the different you can say mistakes or faults that happen in this you can say that in this step of DNA replication. So this is all about DNA replication. Here we have added the slide. It will you read it please carefully. It will be very it will be very helpful for you to write how to this long answer. uh, Again proofreading DNA polymerase जो है ये आपकी proofreading. Here is the summary. First of all, origin of replication is identified. Then RNA primer complementary to DNA template is synthesized. In the third, DNA synthesis is continuous in the leading strand towards the replication fork, and whether DNA polymerase and DNA synthesis is discontinuous in the lagging strand, that is away from your replication fork. Elongation in both strands, the synthesis is from 5 to 3 direction. Then the RNA pieces are moved, the gaps filled by deoxy nucleotides by DNAP, and pieces are ligated by DNA ligase enzyme. Means the RNA primer, they are removed by DNAP. Proofreading is done by DNA polymerase, finally organized into chromatin. Then these are the following enzymes that are involved. This is the summary. Hope so this will be very helpful to you. We will soon come with another topic.